Morning to all.
Good morning, sir.
Good morning, sir.
Good morning, sir.
Morning,
Okay.
sir.
I think the screen is visible, right?
Yes,
Yes,
sir.
sir. Yes, sir.
So, on the um, last class, we have come through some of the basic concepts about the biological databases, and especially the primary nucleotide, the concept of the biological databases, and the major primary nucleotide databases, including GenBank, DDBJ, and EMBL. All these databases were already discussed in the previous session. Now, in continuation to that, we'll be discussing uh, proteomic tools and other related resources, that's database resources available in bioinformatics. Now, I'll share the screen. Uh, Proteomics tools is the uh, topic of today's lecture. And protein sequences, just like that of the the DNA sequences, protein sequences were stored in such databases as single letter codes of the amino acids. So each amino acid will have a single letter representation, and it will be sequentially written or typed to create the protein sequence. So that sequences was stored in the biological databases, and let's see what are the prominent databases in databases for that purpose. That's a protein sequence storage and retrieval. So the first one is NCBI protein resource. As I mentioned in the last class, NCBI they hold many databases other than the DNA sequence database, that is GenBank. They have protein sequence database also. So NCBI protein resources is another database of protein sequences, whereas GenBank is for primary nucleotide sequence. And another one is a protein information resource, or PIR, and that is a identification and interpretation of protein sequence information were stored in PIR database. That was an old database. Now they have actually merged with the next one, that is UniProt. So PIR doesn't have an independent existence because PIR actually contributes to the development of the UniProt knowledge base, that is UniProt database. The universal protein resource, that's including UniProt KB, SwitchProt, TREMBL, UniRef, UniPark. So there are a lot of other related databases which is joined together to constitute uh, this UniProt database, which is now considered as the most prominent database in case of protein sequences. Of course, NCBI protein resources were protein sequence databases. When compared with the UniProt database, their annotations. Annotations means the descriptions attached with that sequence. That will be minimal. So PAR and NCBI resources were normally uh, not depended upon for retrieving protein sequences nowadays. UniProt is the universal standard for retrieving such information. This is the interface of NCBI protein resources. You can see that the only difference is that there is the in the pull down menu protein is selected, so that the search or the query, for example, you are searching for insulin in that search box, it should be against the concerned database, that is protein resource database. So the interface is the same. The query will be directed to the particular database available there, and the result will be from that database only. So NCBI protein resources is one of the resources for retrieving protein sequence information. Now, the second one that is mentioned is a protein information resource, or PIR. Actually, it is compared to the UniProt, this is the oldest one. And this protein information resource of PIR is actually it's an integrated public bioinformatics resource to support genomic, proteomics, and systems biology research and related scientific studies. And this was established in 1984 and by the National Biomedical Research Foundation, that is the NBRF, as a resource to assist the researchers in the identification and interpretation of protein sequence information. Prior to that, these NPRF actually they compiled the first comprehensive collection of macromolecular sequences available in the Atlas of Protein Sequence and Structures, published in 65. That is we already discussed that the first biological database can be treated as a book published, the Atlas of Protein Resources and Structures, published in 65, and that's by Dave. So that hard copy is actually considered as the first resource or first database resource, the first effort to collect all such a structure and sequence related data, and based on which these NBRF or that foundation they developed and established the first PER resource. Now, Dr. Dayov and her research group pioneered the development of computer methods in comparison of protein sequences for the detection of distantly related sequences and duplicates within the sequence, and also and for the inference of evolutionary histories from aligned sequences. And you can compare the DNA or protein sequences and compare them so that the evolutionary relationship between such a sequences, that the organisms which is giving that sequence, that can be compared, that can be related by comparing the sequence information. Then later another group like Winona Barker or Robert Ledley. There are other group of scientists take up that research from Deo after his death in 1983. Then in 1999, Dr. Kathy Shu and later on Georgetown University Medical Center, they take the lead in that research, and then by 2001, the head of the bioinformatics efforts of PIR, they actually, this Georgetown University, they took over the development of that resource, and Dr. Kathy, actually he served in that university until 2001. And for over 4 decades, beginning with the Atlas of Protein Sequence and Structures, that book, then now the PIR has provided protein database and analysis tools and freely available scientific information to the community. So for 4 decades and more, these resources, these PIR resources, are now serving the public with the protein sequences and the protein database. Still, that PIR database is there. You can access at that site, but nobody is actually directly accessing that information because it's already complementing the UniProt database. So there is no point in accessing the data from a less annotated database like PIR. And in 2002, PIR, along with its international partners, that is European Bioinformatics Institute and Swiss Institute for Bioinformatics, that is EBI and SIB. They were also awarded with a grant from National Institute of Health of US, and this collaborative effort of PIR, EBI, SIB, and NIH of US, all collaborated to create the UniProt database, a single worldwide database of protein sequence and function. By unifying the PAR sequence, Swissport sequence, TRENPL database, all the databases were combined together, and now UniProt database, actually it is the best one because it is actually a collaborative effort of all other separate databases. So there are annotations and there are additional information and resources which is attached with the sequences. That is so elaborate. So you can access almost all available information from related with that sequence of protein, their motifs, their functions, and all other related biological information were now attached to that sequence in UniProt. So in 2009, Dr. Wu accepted the Edward G. Jefferson Chair of Bioinformatics and Computational Biology, and there's another position. And okay, it's not about the scientists that we are discussing, it's about the database. So now, as I mentioned, the so, hello, am I audible?
Yes, sir.
Actually, my screen went black. That's why some issue. Okay. Anyway, it's clear, right? Okay. Fine. So the next resource is Expressy proteomic server. So the Expressy proteomic server is Expressy means expert protein analysis system. Is a it's a protein server for bioinformatics, and this is from Swiss Institute of Bioinformatics. That's a SIB. It's a part of European research group, Swiss Institute of Bioinformatics, and which analyzes the protein sequence and structures and the two-dimensional gel electrophoresis or 2D page electrophoresis results. They are also stored in that Expressy system. So other than that, they actually provide a lot of other bioinformatics tools for analysis of the protein and DNA and related sequences. So Expressy has now evolved into a very good resource for, they actually provide, say, a collection of bioinformatics tools for analysis of the proteins, prediction of the structure of the proteins, functions, prediction of the proteins, and they also provide related genomics, proteomics tools, everything. So like, anyway, Expressy proteomic server is actually, it's a server, it's a system, it's a, which is used for analysis of the protein sequence and structures. So you can perform different types of analysis of protein sequences and structures, and even you can access the three-dimensional gel electrophoresis data related with proteins from Expressy proteomic server. And the server functions in collaboration with the European Bioinformatics Institute, uh, that's EBI. Then Expressy also produces the protein sequence knowledge base that is known as a UniProt KB. So UniProt is a protein database, and they also provide a knowledge base of the proteins. So that database is known as a UniProt KB, but that is a Universal Protein Resource Knowledge Base. That is the UniProt KB. And also, that is not, they also have another database that is a SwissProt. Of that, Swiss actually is coming from Swiss Institute of Bioinformatics, and Prot is a protein resource. So Uniprot KB or SwissProt is another source from that Expressy server. And actually, this Uniprot KB or SwissProt, it is a computer annotated supplement. And sorry, it's not. It's the actual database. For example, Uniprot KB or SwissProt is the actual protein sequence database where the sequenced information after biological or experimental sequencing, that sequence will be stored in such databases and they will be annotated efficiently. And that sequences where Uniprot KB or SwissProt database, and that is stored in that type of database. And there is another type of database of Uniprot KB, TREMBL or translated EMBL is the second database in Uniprot KB. So in the Uniprot knowledge base, you have two provisions. One is SwissProt, which actually stores the original experimentally sequenced data, where as the TREMBL component of the Uniprot knowledge base, it is actually the computer annotated supplement, means from the European Molecular Biology Laboratory, that is EMBL DNA sequence database. EMBL, as you know, it's a primary nucleotide sequence database. From that database, the DNA sequence will be taken and it will be that will be translated. Protein sequences can be translated, and the annotation can be generated computationally. So the previous one, it is actually manually curated one. Manually curated and manually verified data is present in Sysbot. Whereas the annotations in TREMPL and the sequences in TREMPL, actually it is computer generated. It is not completely manually verified. So scientifically speaking, you can depend on the Sysbot more because it is actually verified information, actually sequenced information, and manually verified information. Whereas the computer annotated supplement of the protein sequence and the annotations we are present in the TREMPL database. That is actually it can be expanded as a translated EMPL database. Now, between its installation on August 1993 and 5th April 2007, XPC was consulted over 1 billion times. And now within the last 14 years, and the growth of bioinformatics or exponential growth of bioinformatics and big data generation occurred after that time. So now you can see that how many billion times of access has been made from the database, because it may be hundreds of billion times of access may be there for that database during these last 15 years or 14 years. So this is the XPC proteomics server interface, which provides a search box just like any other database. There's a provision for selecting the different types of database, for example, Unicode or Unicode KB, TREMBL. You can select various options, specific databases that can be selected from that pull down menu. And you can give the search result and then query into the search box. So you will get the search result from the concerned database. And also they provide, in the left side panel, you can see there are proteomics, genomics, then structural bioinformatics, systems biology, phylogenetics, transcriptomics. They're all related information. Or if you click on one of these links, you'll be directed to another page which is provided with a lot of tools, maybe 20 or 30, like that. A long list of tools will be there related with that function. For example, if you want proteomics analysis, tools for proteomics analysis will be given in this link. And if you are looking for genomic analysis or the related works, all the related work, tools will be provided in the genomics link. So the XPC system is actually a resource portal, bioinformatics resource portal, which provides different tools for sequence analysis, structural analysis, and function prediction. Everything can be done based on the tools that is. Even structure predictions, three-dimensional modeling of the proteins from primary sequences, that can be done from this XPC system. So this is a main page, and I'm just introducing it because it is having different databases and search provisions, and also there will be a long list of tools which is arranged systematically for your use in the XPC proteomics server. And you can go through that when you are doing the practicals. You can just go through that different resources available in the proteomics server. So this is one of the pages. This is actually the list of the tools. So I don't know whether this page is present now, because as you know, most of the websites will revise periodically every year. And this was a screenshot taken many years back. And actually, the screenshot was from 2011. 10 years back, I had taken this screenshot. And nowadays, actually, they have arranged this in a more systematic way. So this screenshot of this arrangement may not be there. But I'm still using this because here we have a single list of different resources like protein identification and characterization. If you are looking for this purpose, these are the tools available. Okay, fine mode, fine pet, mass code, pet marker. So these are different specific tools for specific tasks related with the protein identification and characterization. And if you are looking for identification and characterization with the MSMS data, mass spectroscopy data, then you can use these tools. So if you have the mass spectroscopy and you want to identify the protein based on that mass MSMS data, then you can use this type of tools. Then identification of isoelectric point and molecular weight. So different types of characterization of the protein is possible with these type of tools. And similarly, you can browse through these links and see what are the different type of analysis which is provided and the related tools which is made available in that link. It can be accessed from that website. So this is actually a search in the Uniprot knowledge base. You can see that this is Uniprot website. This page is say you have to get a protein sequence from Uniprot. You have two options. One is you can select the database Uniprot from this pull-down menu in Expressy. If you select Uniprot as the database in this pull-down menu and give the search term, say insulin in this one, and search, then it will run the search in the Uniprot database, and the result will be returned, or the page will be completely redirected to the Uniprot search results. If you select the Uniprot database, the result will be available from this page because the search will be, or the query will be directed to this page and result of this particular search query. For example, it will be the same as the previous query which is submitted here. By selecting the Uniprot and giving the query here, that will be the same as the query which is given here in the original Uniprot site by selecting Uniprot knowledge base or Uniprot KB. So this same database is actually provided with the Sysprot also. So the search result will be the same. So you can give the same query here directly in the Uniprot KB. So either you can use the Expressy resource or you can directly go to Uniprot and give the query of the concerned protein. So this is the home page of Uniprot. And this is some statistics related with the number of entries present in the Uniprot. And you can see that by 2019, about more than 140 million entries were there. 140 million is 14 crores of sequence entries were there in Uniprot KB database. So now, during the last 2 years, it might have grown further. Okay. So that is the actual capacity or almost size of that Uniprot database. And of course, there is a TR-EMBL, translated EMBL. It is not completely manually sequenced or experimentally determined sequenced. So because it is a Uniprot KB translated EMBL database, so it contains manual computer annotations and predictions also. So the original CD sequenced one may be much less. Okay. So Uniprot actually they provide protein knowledge base consists of two sections. The first one is a Sysprot, which is manually annotated and reviewed. TR-EMBL, which is automatically annotated and not reviewed. So this includes complete and reference proteome sets. So that's Uniprot KB. Then UniRef is a sequence clusters used to speed up the sequence similarity searches. So UniRef actually they are references sequences or sequence clusters. So that type of reference sequence were usually required for speeding up the search, and that's a UniRef. UniPark is the sequence archive used to keep track of the sequences and their identifiers. So there are other supporting data in the Uniprot: literature citations, taxonomical information, keywords, subcellular allocations, cross-reference databases, biological information, biological activity, and a lot of other information related with that concerned protein that is also annotated, provided by that Uniprot. So see this example in which the Uniprot database is searched with the query insulin, and you have a lot of results available. And you can see that the entry name is from human, three entries from human, another one bovine, another from human, then from mouse, rat, sheep. So from different sources, we have different protein sequences. And maybe that's protein gamma, or which peptide, or beta and alpha, epsilon, which segment is actually there. That is also marked. The protein name is there. Then the source organism is shown, scientific name of the source organism is shown here. This is the accession number or unique accession number of the protein in the Uniprot database. And at the end, you can have the length of the sequence. And if you are trying to download a sequence from this site, you have to to see the description, whether it is the complete or intended protein or the complete protein or the target protein itself, or some related protein or some binding site or some other inhibitor protein. Everything will be listed if you are typing insulin. So you can verify whether the listing is of the concerned protein and whether it is matching the concerned length of the protein, desired length of the protein, and if it is from the concerned organism or not. And all these things can be verified before going to the details and downloading the protein. So this is the actual link to download the protein and see the information. So once you click on any of this link, for example, here the first link can be opened by clicking on this one. And once you open it, you can have the detailed annotations or descriptions on that one. So here you can see that this is the ID which is open, and the ID number and source organism is marked here. And the version number is also, and the publication year is 2010, and this is the 116th version of the revision. And the recommended name of the protein, and what the class is actually it's a protein kinase, C inhibitor protein 1. Then similar descriptions of the protein is there. Then organism, it is a Homo sapiens of human. And similarly, you can see on the right side where there is a scroll bar, and the size of the scroll bar is very small. That means the page is a very long page. You can scroll down the page, and there will be a lot of information, as I mentioned in the previous slide, like literature citations, taxonomical information, keywords, subcellular locations, and biological activities, cross-reference to databases, like links to other databases, etc. And all these things will be available if you scroll down to this page. And it's better you go and visit this page, Unicode, and search with any query anytime, and open one of the citations or one of the retrieved sequences, and see the nature of the annotations or the descriptions. All other related biological information related with that protein were given in a systematic and in a very classified way. So that is the Unicode annotations. So okay, that's about Unicode. Now this is another option, as I mentioned. So this is the Expressive Proteomics server, and in the Expressive Proteomics server, you can select the Unicode KB database. Instead of directly searching in this Unicode KB, you can use this option also. You can pull down, select from the pull down menu. You can select the Unicode KB database and give the same query like insulin here, and the result will be the same page that's previously discussed. So that's that's about the details about the protein sequence databases, like NCBI protein resource, then protein information resource, then the Uniprot knowledge base with very detailed annotations on the protein sequences and the related function and other information, related informations. Is there any doubts in this section? Hello?
No, sir.
No. So let me proceed with that. Then the next one is a protein structure database. Protein structure database, the prominent one is a protein data bank. And this protein structure database, they contain three-dimensional structure, basically three-dimensional structure of the protein, or files containing the three-dimensional structure of the protein. So this protein data bank is actually maintained by the organization Research Collaboratory for Structural Bioinformatics. This is also known as RCSB. So the website will be www.rcsb.org. And previously the PDB, www.edb.org, that will be redirected to their homepage, that is rcsb.org, and it is the same. So that provides the database of a protein data. Then there are other related structural database like protein model portal, PMP. There is a meta database that combines several databases of protein structure models. And it's a another database. Then CAT protein structure classification. Structure classification database, not structure. Classification of the structures is provided in the CAT database. Scope is another structure classification of proteins. Then Swiss model server is a repository for protein structure models. So this risk model server is basically used for a homology based. That's a technology that we will discuss in the next sessions. So there is a modeling approach in which you can model the three-dimensional structure of the protein based on the primary sequence. And that service is provided free of cost by the Swiss model server of the Express. Now there is a mod-based database of a comparative protein structure models. That's another one. So there are some specialized databases, but the main database that is used worldwide by almost every bioinformatician is a protein data bank, which provides the structure of all the available all the determined structures and even predicted structures of the proteins were made available in the protein data bank, freely available to the scientists worldwide. So this is the homepage. Again, this screenshot is taken in 2013, as shown in the timeline. It's taken in 2013, and this might have changed its look and feel, appearance, but the basic approach of the search or query is the same. It provides a query box in the front end itself where you can give the PDB ID or the unique ID of the concerned structure or its name or other related information to retrieve the concerned structure information. So this PDB, when you compare the size of this database, you can see it's up to 2013 only. 2013 only. So this is yearly growth of the total structures, total number of structures. This is about 90,000. And this is about only 10,000, this government. Okay. So this is a year and the actual structures, this is a actually determined structure based on X-ray crystallography and other things, total structures. And this is yearly growth. This is the total structures. This is a yearly growth. So in the single year, what is the quantity that is added to the database? So that is shown here. This is the available total structures in every year. So every year it is growing. And this will be up to 2013, but you can see the same graph and extrapolate it to further. So this is a current position holding up to maybe it's 2019. 2019 or 20 such is there. So in this case, you can see about the X-ray crystallography based structures of proteins. This much. Nucleic acid three-dimensional structures and other related structures or protein other related compounds or some other ligand bound compounds and other things. So total structures determined based on the technique X-ray crystallography is almost 80,470. NMR based structures were also there, that is less than 10,000. Then electron microscopy, then hybrid methods and other methods. So there are various methods for the actual determination of the three-dimensional structures. And the prominent one is the X-ray crystallography. Then comes the NMR. So based on all these techniques, these are the number of structures that is actually added to the database. So you can see about 69,919 structures in PDB have a structure factor file, and 7,260 structures in PDB have an NMR restraint file. So these are additional related information about the structural determination. So because if somebody else is using that same structure, it is better how the structure is determined. So such type of detailed data related to the structural determination is also available in this PDB database. Now, this one is a screenshot of the search or query result. So if you, for example, the query is for insulin, and in the PDB database, I searched for the insulin, and the query result will be something like this. So there will be a small visualization or graphical visualization of the structure. And there are some icons for seeing the structure. For example, if it is a Java-enabled browser, previously, if you click on this point, that will be opened. The structure will be opened in the three-dimensional model will be opened in the browser itself. And also, you can see the sequence and other structural information, etc., by clicking on these links. Now, the brief description about the protein and its release date and the chain or the here we have only one chain, A chain. Sorry, A and B chains are there. So both chains and their nature, etc., are mentioned here. And some brief description about the authors, etc., that are also provided here. The A chain is having 21 amino acids, and B chain is having 26 amino acids. So that type of basic information is provided there. And if you click on and also you have to look into the title. This is a semi-synthetic, highly active analog of human insulin. Okay, and the semi-synthetic, highly active analog of human insulin. And sometimes the result will be insulin inhibitor protein or insulin binding protein or insulin like protein. Anything may be listed there, but you have to look in the description and see that it is actually the concerned the structure of the this is actually the structure of the concerned protein that you are looking for, because in such a computational queries everything in the title with the concerned term will be listed. So you have to take into consideration that there is a possibility that most of the query results may contain unwanted items like insulin-like, insulin inhibitor, insulin receptor, etc. So if you want to download the structure of insulin, it should be insulin stuff, not the related ones or binding proteins or etc. And also there are some provisions here for filtering. Sorting results are there, and also maybe if you look into the side menu, you can NMR, for example, on the left side panel, we can have electron microscopy, NMR, etc. So if you click on that NMR, it will be all the NMR related structures will be listed, excluding the others. So there are different filtering options which will be available in the database. So this is the basic understanding of a query result. So you have to determine which one to be dependent on, which is based on these primary informations about the sequence length and the name of the title, from the title, from the length of the sequence, and other descriptions. So once you click on this link, that the file will be open, or the detail of the detailed annotations with the protein will be open. So once you open that one, say here is a title and or the article in which it is published, and the authors and the name of the publication, or the proceedings of National Academy of Science USA, 1966. So that detailed information related to the publication of that structure is there. Now the molecular description. Now this is what's shown in the search results. Now the source of the protein, then related other PDB entries, and all other related information will be provided as annotations in the query results. So that is about protein data bank and retrieval of the sequences. And of course, you have practical sessions which will be unlocked today itself. And there you can have a video demo on the method of downloading that sequence from protein data bank. Sorry, the structure from protein data bank. You can perform that in your own computer. Now this is about the Swiss model server. So this is an automated protein structure homology modeling server, as I mentioned. So from the primary protein sequence, you can, using the Swiss model server, you can model the three-dimensional structure of the protein. So that's about the Swiss model server, which is actually a modeling server based on homology approach, or it's a homology modeling approach. Now then, there is other protein-related databases like Interpro, then InterPro is an integrated resource for protein domains and functional sites. ProSite, that is a database of protein families and domains. Then blocks is another database, blocks database. Protein blocks were stored in that protein blocks database. Gene3D is a structural and functional annotation of protein families. So structural and functional annotation or description of protein families were present in Gene3D database. Then Bandar is for a functional classification system. PFARM is a protein families database. Then Prodome is for a protein domain, is a protein domain database. So it's a motifs, domains, classification, then function and structure. Every detailed aspects of proteins were stored in such specialized databases. So these are some of the list of protein related databases. There are again some other list, superfamily representing protein, all protein. It's a library profile of a hidden Markov model based, that's HMM representing all protein sort of non-structures, superfamily informations. Then string is a very useful database. Maybe I will be showing some more slides on that one. String is actually, it's a search tool for retrieving of interacting genes of proteins. So I find it very useful to get a better understanding on the protein interactions. So string is a very useful search tool, database search tool. Then when coming down, you have MIPS, that is Munich Information Center for Protein Resources. So I will be selecting some of them. For example, Interpro, then ProSite, blocks, string, and MIPS, etc. Some of them, the highlighted ones, will be discussed in the subsequent slides. So this string, as I mentioned, it is known and predicted proteins. That protein-protein interaction, known and predicted protein-protein interactions were retrieved using string search. So string is actually protein-protein interaction prediction. That if it is already known interaction, that will be also added into that one, and even it predicts the possibility of protein-protein interactions. So here you can say in the search box I have given a query term like myeloperoxidase, which is an enzyme in the WBC, as you know, which prevents microbial infections. Okay, I just search for myeloperoxidase. So you will have a query result, and as I mentioned, this screenshot is also a very old one, and so this screenshot might have changed, or the query result might have changed in the recent years, based on depending upon the revision in the website design. So as I mentioned, this is a very old screenshot. So in the screenshot and the query results on myeloperoxidase has returned various results like from different sources. So this first one is from Homo sapien. This is from other organisms. From different organisms, the myeloperoxidase, that gene were listed here. So I have clicked on this myeloperoxidase from Homo sapiens, and if you select any of them, then the MPO is actually positioned at the center. That's myeloperoxidase is positioned at the center, and all other related proteins, or which may have interaction. It also it predicts the interactions also. So all proteins with proper interaction or predicted interaction will be shown, and their interconnection. So what proteins will be interacting with the or which protein is interacting with which protein will be shown with a graphical representation in the string database. So this next one is about ProSite. ProSite is for protein domains, families, and functional sites. So the information related with the protein domains, protein families, and their functional sites were stored in the ProSite database. And normally such database are advanced databases. They provide some graphical interface or graphical representation of the protein families, domains, etc. So the next one is Munich Information Center for Protein Sequence, that's MIPS. It is hosted by the Institute of Bioinformatics and Systems Biology, and it's a part of German Research Center for Environmental Health. Its main focus is the genome-oriented bioinformatics. In particular, the systematic analysis of genome information, including the development and application of bioinformatic methods in genome annotation, expression analysis, and proteomics. So it's basically a German-based research sector, and their function is development and application of bioinformatics methods in genome annotation, expression analysis, and proteomics. MIPS supports and maintains a set of generic databases as well as systematic comparative analysis of microbial, fungal, and plant genomes, etc. So this is a German-based database which stores gene information, their annotations, and comparative analysis results of microbial and fungal and plant genomes. So this is one of the this is the homepage. It's also an old one. That homepage of MIPS, and there, for example, you can access the entire Orisa Sativa database. So the Orisa Sativa database is actually specialized database for the entire genome of the Orisa Sativa or the rice and the related informations. So they provide such a specialized database, rice genome database. That's Orisa Sativa database, and in which you have a graphical representation of all the genes, all the chromosomes which is present, and you can access the information and browse the information and the different genes and locations or the loci in the concerned chromosome with a graphical browser. So this is possible for with the MIPS. So you can see that if you open one of the chromosomes, this is chromosome 3 as this is shown here. This is a chromosome 3, and that molecule is open. And if you select one of the regions in this region, and in the highlighted, for example, in the start point here, if you select a region from the top, then subsequent selection will be available in the next region, and in that particular selected region, the detailed descriptions about the different genes and loci present in the concerned gene is available in the graphical interface. So that's about structure-related databases. Now the next one is about metabolite databases. Hello, am I audible?
Yes, sir.
Okay.
Yes, sir.
Actually, I lost my power supply. That's why. I want to confirm that everything is working as normal. Okay, the next one is, as I mentioned, this is a metabolite database. One example is ECOSIC. This is of Escherichia coli, special database for Escherichia coli. And ECOSIC is a scientific database for the bacterium E. coli, specifically that's a ketol, MG1655. So it's a very specialized database for that particular E. coli. Okay, and this is a scientific database for the bacterium, and this project performs a literature-based curation of the entire genome and the transcriptional regulation, then transporters and metabolic pathways. So all these things related with this particular organism, any information that is the genome related, transcription regulation or transporters or metabolic pathway. Also, any such related information generated on this particular organism will be added in this database. And that will be based on literature-based curation. For example, if you are performing a research on this organism, and if you are publishing a result based on that research, then that is a literature that is published. Based on that published literature-based information, that will be added, or this information will be updated, and it is manually curated and it will be added into the database. So it's actually a specialized database which is used for say E. coli K12 strain. So the ECOSIC is a bioinformatics database that describes the genome and the biochemical machinery of this particular strain of E. coli 12. And the long-term goal of this project is to describe the molecular catalog of the E. coli cell as well as the functions of each of its molecular parts. So if you add all such information on a particular organism into a single interface or into a single database, and it facilitates a system level understanding of the E. coli. So if you can define every metabolic aspect, every pathway, every enzyme, and their interactions, then that is actually a systems biology approach in which a computational model of an E. coli cell can be created if you understand every aspect of a cell. It's not E. coli. It's basically a systems biology approach in which you will have a better understanding. With increasing understanding, you will add all more and more information into that one. Uh, and if you can define the entire metabolic system or entire metabolic pathways of a particular organism, then you can simulate an E. coli cell in a computerized environment. So you can say what will happen if you mutate one gene or if you add some metabolite additionally, if you have some depletion in a metabolite, what will happen to the next of the system or how the cell will respond. That's a type of system modeling is possible if you develop such a database or such tools. So the long-term goal of this project is to develop such a system-level understanding of the E. coli, which may facilitate further development in the systems biology, or such a modeling of a computational model generation of E. coli. ECOSIC is an electronic reference source for E. coli biologists and for biologists who work in the related microorganisms. So this can be considered as a model for other organisms also. So the next one is KEG, that is a Kyoto Encyclopedia of Genes and Genomes. It's also a very useful database. This is maintained by Japan. Since 1995, this KEG database has been developed developed in the laboratories of Japan. That's then it's at Kyoto University, and also got funding from different Japanese funding agencies. So in the KEG, they have a lot of other, for example, main entry point of KEG web service, that is KEG2, then KEG pathway, KEG module, KEG drug. So for a very specific, very specialized purpose, you have different tools available. So if you are looking for pathway information, you can select pathway information. If you are looking for some disease modules, then you can have a module. And if you are looking for some drug and related information, KEG drug is the option. So if you are looking for entire genomes in the KEG, then KEG is option. So what I find interesting is KEG pathway. KEG pathway will give you a better understanding on metabolic pathways. Okay. So here, if you open the table of contents, you can have systems information, genomic information. It is categorized as chemical information tools, genomic information tools, systems information tools. This is also a previous screenshot. It might have changed in the current years. Anyway, basic concept is that they provide many entry points with specialized purposes. Okay. So here I have access to this KEG pathway, and KEG pathway is used here. It is searched with the term insulin. So it will return a lot of entries for insulin, and I have opened one of them. So if I open one of them, then insulin signaling pathway is actually shown here. So if you have any enzyme of interest or any protein of interest, that can be given to the query. And if it is listed in the pathway, or wherever that protein is interacting in any metabolic pathway, that will be listed. And if your interesting metabolic pathway is listed there, you can open it. Then see, this is an insulin signaling pathway that is opened here, and description about the pathway is provided here. Then the pathway, a screenshot of the pathway, a miniature form is shown here, and related other informations and publications are given here. This pathway diagram can be expanded, and so it will look something like this. So the entire insulin signaling pathway is now available to you, so that you can have a better understanding on insulin signaling pathway and its various other various other enzymes that is involved, so that even you can try to do some research ideas related with that insulin pathway based on this one. Okay, so usually when you are trying to find some research gaps, or you are trying to interpret something based on some insulin pathway, so such type of overall idea about the interacting metabolites and other uh, items will be very useful. So that is available in such a metabolic pathway, reference pathways where all can be retrieved from the KEGG interface. There is another tool, that's a BioCarta pathway, which is also another, it's not a part of KEGG, it's a separate pathway tool. So it's available from biocarta.com. So they also provide some kind of metabolic pathway information. So if you have some interesting protein or enzyme, you can give the search name, and if it is listed or if it is included in any of the pathways, that pathway will be listed. You can go have a graphical representation of the pathway from that BioCarta pathway. So the next one is a secondary database. So the secondary database, some examples are DB browser, one example. So here Prince is a compendium of protein fingerprints. So it's actually it compiles it gets the information from various sources, and then that protein fingerprints will be stored in this Prince database. So a fingerprint is a group of conserved motifs used to characterize the protein family, and its diagnostic power is refined by iterative scanning of a Swissprot or TRNBL composite. So this is actually these fingerprints were actually conserved motifs of which are characteristic of a particular protein. So that type of information. So conserved motifs of proteins were grouped as fingerprints, and they were stored in the Prince database. So that's another one. Then ProSite, as I mentioned, it is a database of protein families, domains, and functional sites. Then blocks is another one. So this is also another type of database which stores protein blocks and multiply aligned gapped segments corresponding to most highly conserved regions of the proteins were stored in the blocks. Now, interpro protein sequence analysis, that is another one. Interpro protein sequence analysis and classification is available from EMBL, or European Molecular Biology Laboratory, and that's EBI. So Interpro protein sequence analysis and classification database is another tool. Then there are database search engines or meta search engines, and we have discussed these ones as the tertiary database services. So meta search engines are the search engines or tools that is available to search across various databases were another category. So as we have mentioned here, we have different types of databases. It is not the complete picture because there are maybe hundreds of databases out there, but these are the prominent databases in NCBI. PubMed for literature, for text electronic journals, then nucleotide sequences, that is GenBank, protein resources for protein sequences, then genome sequences, online Mendelian inheritance in man database, 3D structure database, taxonomical information database. So various other database, GST, EST, that's express sequence tags. So there are a lot of other databases out there. So Yendrus is a search engine that is provided by the NCBI to search across all these databases, and a single query that is given to the Yendrus will search within all these databases and retrieve the results simultaneously. And the hits in each database will be listed against the concerned entry. Here you can see one of the examples. Say, in this case, I have searched this database. Sorry, this is the interface of the NCBI address where we have a search box, and the possible search database were also listed here. Welcome to address to cross database search. So these are the available databases, some of them. And in the next page, you can see, say, I have searched for insulin in the address interface, and PubMed, which is a biomedical literature and citation abstracts database. PubMed is having about 279,542 articles related with that one. The PubMed Central is a free full text. There's more than 1 lakh. Now, this is about books. 512 books is there in the books database. Online embedded in HSS man, that is 924 entries related with insulin were present in that database. And this is the GenBank, that is a nucleotide database. That is a 22,250 entries related with the insulin were there in the nucleotide sequence database of GenBank, express sequence database. So you can see every type of specialized database will have different entries, and it will be listed against that concerned database in the end search. So this is the concept of the meta search or meta search engine, which self-check those various databases. And if you have this result, then you can look into various databases, and the interested data can be retrieved from the concerned database by clicking on that one. And only the result from that database will be retrieved from the site. If you are clicking on EST, so that means your query is against the EST database in the main page. If you are clicking on this one, it is equivalent to the query of insulin against nucleotide database. So this is another one, that is SRS, which is previously mentioned, sequence retrieval system. And this is from European Molecular Laboratory, that's EMBL. EBL search is done with SRS, and they also provide the search provision, and their search results will be something like this. So research results against genomes, nucleotide sequences, protein resources, macromolecular structures. So different entries were listed in the search results. And the interested area, for example, if you are looking for protein families related with insulin, then you can click on this one in the SRS search result. So then DDBJ, they have GetEntry as the search service, cross-platform database search service. You can use that service for search results. So this is the DDBJ search results. Compared to the NCBI or the NCBI services or ENDRA services, the SRS and these GetEntry services were not that much effective because their data representation, you can see that these data representation will be not up to the mark if you are comparing that ENDRA results, or the additional resources available in the additional information available in the NCBI is, I find, at least I find it more useful. So preferably, if I am looking for to retrieve some information, I will be generally visiting the NCBI, not the other two. Okay, thank you. Any questions? Hello?
No, sir.
Okay. Then let me start it.
Okay, sir.
Okay.